plant virus harnessed to fight cancer. New immunotherapy breakthrough. UCSD study. Imagine a virus, not from a lab, but from a humble cowpea plant, holding the key to unlocking our body's natural defenses against cancer. That's the astonishing discovery made by researchers at the University of California, San Diego. They found that the cowpea mosaic virus, CPMV, can reprogram our immune system to fight cancer, while other similar viruses fall short. But here's where it gets controversial. The study, published in Cell Biomaterials, reveals that CPMV triggers both the innate and adaptive immune pathways, priming our immune system to recognize and fight cancer cells. Unlike other plant viruses, CPMV stimulates an anti-tumor response, making it a promising candidate for cancer immunotherapy. The research team, led by Nicole Steinmetz, PhD, and Anthony Omelay, injected CPMV directly into tumors and observed a broad immune response, drawing various immune cells into the tumor microenvironment. This activity not only fights existing tumors, but also primes the immune system to recognize and eliminate metastatic tumors. But how does CPMV do this while other viruses fail? The researchers found that CPMV's RNA persists longer within immune cells and activates a key receptor, toll-like receptor 7, TLR7, which is essential for triggering antiviral and anti-tumor responses. In contrast, another virus, cowpea chlorotic mottle virus, CCMV, failed to activate TLR7, explaining its lack of therapeutic effect. And this is the part most people miss. While CPMV doesn't infect mammalian cells, it's recognized as a foreign pathogen, triggering a cascade of immune activity. It doesn't kill cancer cells directly, but instead primes the immune system to recognize and attack them. This tumor-agnostic efficacy has been demonstrated in multiple mouse models and even in canines with cancer. But here's the catch. The researchers caution that their findings are still preclinical and require further validation in human systems. They're refining the formulation, dosage, and treatment protocols as they prepare for early clinical trials. The potential is vast, but there's still much to learn. As the team moves forward, they're inviting us to join the conversation. What do you think? Is this the future of cancer treatment? Or are there potential risks we need to consider? Share your thoughts in the comments below. The beauty of the mammalian immune system lies in its ability to recognize pathogen-associated molecular patterns, PPAMVs, from viruses across different kingdoms. Could CPMV be the key to unlocking a new era of cancer immunotherapy? The next chapter is yet to be written, but the possibilities are truly exciting. Plant virus cancer cure. CPMV immunotherapy breakthrough explained. Imagine a world where a simple plant virus could revolutionize cancer treatment. This is not just a dream, it's becoming a reality with the cowpea mosaic virus, CPMV, a virus that typically infects black-eyed peas, which is emerging as a promising, cost-effective cancer immunotherapy. Researchers are delving into the reasons behind its remarkable effectiveness. In a groundbreaking study published in the journal Cell Biomaterials, a team of chemical and nanoengineers from the University of California, San Diego explored how CPMV stands out from other plant viruses by effectively activating the immune system to identify and attack cancer cells. Preclinical trials have shown that CPMV exhibits strong anti-tumor properties in various mouse models and even in dogs suffering from cancer. When injected directly into tumors, CPMV therapy mobilizes innate immune cells such as neutrophils, macrophages, and natural killer cells into the tumor microenvironment where they work to eliminate cancer cells. Additionally, it stimulates B cells and T cells, creating a long-lasting immune memory that not only helps eradicate the targeted tumor, but also prepares the immune system to seek out and destroy metastatic tumors that may appear elsewhere in the body. Nicole Steinmetz, the Leo and Trude-Zillard Chancellor's Endowed Chair in the Aiso Yufeng V Family Department of Chemical and Nanoengineering at UC San Diego, And the study's lead author expressed her fascination. It is intriguing that CPMV, unlike other plant viruses, can trigger an anti-tumor response. Anthony Omo, a PhD student in Steinmetz's lab and the first author of the study, added, "Our findings reveal that even though human immune cells do not get infected by CPMV, they still respond to it. This response reprograms them into an activated state, effectively training them to recognize and eliminate cancerous cells." A pivotal question in the journey to translate CPMV for human cancer patients is, what exactly makes this plant virus so effective against cancer? To answer this, Omo, Steinmetz, and their colleagues at the National Cancer Institute's Nanotechnology Characterization Laboratory conducted a comparative analysis of CPMV and the cowpea chlorotic mottle virus, CCMV, a similar plant virus that does not show anti-tumor effects when injected into tumors. Both viruses create nanoparticles of comparable size and are absorbed by human immune cells at similar rates. However, their effects diverged significantly once inside the cells. The research team discovered that CPMV triggers the production of type 1, 2, and 3 interferons, proteins known for their anti-cancer capabilities. Omo pointed out, this is particularly noteworthy because some of the earliest cancer immunotherapy drugs were recombinant interferons. In contrast, CCMV leads to the production of pro-inflammatory interleukins that do not contribute to effective tumor clearance. Another critical difference lies in how the RNAs of these viruses are processed within mammalian cells. CPMV RNAs have a longer lifespan and are transported to the endolysosome, where they activate toll-like receptor 7, TLR7, a vital component in initiating antiviral and, more importantly, anti-tumor immune responses. Conversely, CCMV RNAs do not reach this activation stage. Moreover, CPMV presents the unique advantage as a budget-friendly immunotherapy option. Unlike many other treatments that require intricate and expensive manufacturing processes, CPMV can be cultivated through molecular farming. Omo explained, it can be grown in plants using sunlight, soil, and water. The research team is now focused on advancing CPMV toward clinical trials. Steinmetz emphasized the significance of their findings. This study sheds light on the mechanisms behind CPMV's effectiveness. We are committed to taking the necessary steps to ensure that we select the most potent candidate for achieving both anti-tumor efficacy and safety. This is an exciting time, and we are ready to transition this research from the laboratory to clinical trials. This research receives support from various organizations, including the National Institutes of Health, NIH grants R01 CA224605, R01 CA253615, R01 CA2746404, the American Cancer Society, the FM Kirby Foundation, Inc., the Mission Boost Grant, MBGI 2310324440MV, the Shaughnessy Family Fund for Nano-Immunoengineering at UC San Diego, the San Diego Fellowship Fund, the Alfred P. Sloan Foundation's Minority PhD Program, and the Frederick National Laboratory for Cancer Research, funded by the National Cancer Institute, part of the NIH. Plant virus destroys cancer cells. Revolutionary immunotherapy breakthrough explained. What if the key to defeating cancer lies in a humble plant virus that infects black-eyed peas? It sounds like science fiction, but researchers are uncovering how this tiny invader might hold the power to revolutionize cancer treatment. Imagine a therapy that's not only potent, but also dirt cheap to produce, using sunlight, soil, and water as its raw materials. That's the promise of the cowpea mosaic virus, CPMV, a discovery that's sparking both excitement and debate in the medical community. But here's where it gets controversial. Could a virus that doesn't even infect humans be the secret weapon we've been searching for in the fight against cancer? Let's dive into the science behind this groundbreaking research and why it's raising more questions than answers. In a recent study published in Cell Biomaterials, a team from the University of California, San Diego, UCSD, has peeled back the layers of how CPMV, unlike its plant virus cousins, sparks a powerful immune response against cancer. This isn't just about a virus attacking tumors. It's about reprogramming the body's own defenses to recognize and destroy cancer cells. The study, titled Comparative Analyses for Plant Virus-Based Cancer Immunotherapy Drug Development, reveals how CPMV isn't just effective, It's uniquely effective. Preclinical trials in mice and even canine cancer patients have shown that when injected directly into tumors, CPMV acts like a biological alarm bell, recruiting immune cells like neutrophils, macrophages, and natural killer cells to the tumor site. These cells then launch a coordinated attack, eradicating cancer cells and leaving the immune system primed to hunt down any metastatic threats lurking elsewhere in the body. But what makes CPMV stand out from other plant viruses? The answer lies in its ability to trigger a cascade of immune responses that other viruses can't replicate. For example, CPMV activates type 1, 2, and 3 interferons, proteins long known for their anti-cancer properties. In contrast, a closely related virus called cowpea chlorotic mottle virus, CCMV, stimulates pro-inflammatory interleukins that don't translate to effective tumor destruction. This difference isn't just academic. It's a critical clue in designing better immunotherapies. As lead author Anthony Omole explains, even though human immune cells aren't infected by CPMV, they still respond to it by flipping into an activated state. This reprogramming teaches the immune system to detect cancerous cells with remarkable precision. The magic, however, goes deeper. Inside human cells, CPMV's RNA lingers longer and travels to the endolysosome, where it activates toll-like receptor 7, TLR7. This receptor is a linchpin in triggering antiviral and anti-tumor immunity. CCMV, on the other hand, fails to reach the activation threshold. Think of TLR7 as a security guard who only responds to a specific key. CPMV has the right key, while CCMV doesn't. This discovery opens the door to engineering other viruses or nanoparticles to mimic CPMV's success. And here's where the cost-effectiveness angle becomes even more compelling. Unlike many immunotherapies that require expensive lab manufacturing, CPMV can be grown in plants using molecular farming techniques. As Omole puts it, it's like farming tomatoes, but instead of fruit, you're harvesting a drug. This scalability could democratize access to cutting-edge cancer treatments, especially in low-resource settings. But is this the future of medicine, or are we underestimating the risks of using a plant virus in humans? Critics might argue that long-term safety studies are still needed, and the potential for unintended immune reactions can't be ignored. The UCSD team is already racing to translate these findings into clinical trials. As senior researcher Nicole Steinmetz notes, the study's insights into CPMV's mechanism of action are paving the way for selecting the most potent candidate for human trials. Yet, the road ahead is fraught with questions. How will the immune system react to repeated doses? Could CPMV's effects vary across different cancer types? And what about the ethical implications of using a virus that thrives in crops we eat? These are the debates that will shape the next chapter of this research. So what do you think? Is CPMV the unsung hero of cancer immunotherapy? Or are we chasing a silver bullet with hidden flaws? Share your thoughts in the comments, because the future of medicine might hinge on this very conversation.